Good morning, class, and welcome to week 2 of AMP1, or if you are in a summer program, then this is week 1, part 2. So today we're going to talk about the cellular level of organization. So cells are the building blocks of all plants and animals, and they come from the division of pre-existing cells. Cells are the smallest units that perform all vital functions. So I want to reiterate that. That's an important point, that the cell is the smallest unit that performs all vital physiological functions. And each cell maintains homeostasis at the cellular level. So we have homeostasis at the level of the cell, at the level of the tissue, at the level of the organ system, and the organism. And this is through the coordination of many different types of cells. We have two different types of cells in the human body. Sex cells, which are our germinative cells, are reproductive cells. In male humans, that is the sperm. In females, it is the oocyte that develops into the egg. You're going to learn about that in AMP2. But in AMP1, we're going to be discussing somatic cells, which are all the other body cells aside from sex cells. Our cells all look very different, but they contain a lot of the same major parts. So we're going to talk about cells right now in a very general terms. So the plasma membrane is the outside of the cell. The cytoplasm is the inside of the cell. And then the nucleus is the large central portion of the cell. The plasma membrane forms the outside of the cell. It's a division between what's outside the cell in the external environment and what's inside the cell in the internal environment. The fluid that's outside of the cell is called the interstitial fluid. That's fluid that is within tissues in between the individual cells, is the interstitial fluid. Plasma membranes are what we call a selective barrier. That means that some things are allowed to pass freely and other things are not. They require active movement in and out of the cell. So the membrane is what decides who and when gets into the cell and gets out of the cell. It also plays a key role in communication between different cells and their external environment. The plasma membrane has multiple functions. It's a physical barrier that isolates the inside of the cell from the surrounding environment. It regulates exchange with the environment. So like we were just saying, it decides what ions can come in and what nutrients can come in, and then it eliminates waste products and other cellular products. So if your cell is producing a hormone, that hormone has to get released across the plasma membrane. It is sensitive to the external environment, so it's always monitoring the signals in the external environment through like chemoreceptors, so receptors that are looking at what chemicals are in the area. And then it also provides structural support to anchor the cell within tissues or sometimes to move the cell. Some of our cells are mobile, and so it keeps the structure of the cell intact. The membrane is a lipid bilayer. So lipid is the term for fat. Bilayer is just what it sounds, 2 layers. So it's 2 layers of these phospholipids, which you can see here. So the phospholipids have hydrophilic heads. Hydrophilic is water-loving. So their heads, which are, you can see these orange circles here, those are the heads. So hydrophilic heads and hydrophobic tails. So the tails you can see in the center are going toward one another, and the heads are on either side. So the outside of the cell is a more watery environment, as is the inside of the cell. But so the water-loving hydrophilic heads are on the outside, and the inside of the plasma membrane. And then the hydrophobic fatty acid tails are on the inside facing one another. I always think of a cell membrane like a peanut butter sandwich, where the hydrophilic heads are the bread on either side. And so if you think about what would happen if you poured water onto a slice of bread, the bread would get all soggy and it would absorb that water. Whereas the hydrophobic tails are like the peanut butter. So if you had a layer of peanut butter and you poured water on it, that water would bead up and run off of the peanut butter, right? Because the peanut butter is fatty and therefore it's hydrophobic. It's not, if you mix oil and water together, the oil will not dissolve into the water. It will ball up and bead up because it's hydrophobic. So our peanut butter sandwich, we have 2 layers of bread, which is the hydrophilic heads, with our hydrophobic peanut butter in the middle. And the plasma membrane is a barrier to ions and water-soluble compounds. So water-soluble compounds cannot pass through this center area where the hydrophobic tails are. Only fat-soluble compounds can diffuse through a plasma membrane. There are many different types of proteins within and attached to our plasma membranes. Integral proteins are just what they sound. They are integral to the functioning of the plasma membrane. And if you were to remove them, you would damage the membrane. So these are called transmembrane proteins. So they're proteins that go all the way through the membrane. And you can picture that if you were to pull that out, you would leave this hole and a damaged area in the cell membrane. Peripheral proteins are bound to the outside of or the inside of the surface of the membrane, and they can be peeled off without really damaging the membrane itself. I think of peripheral proteins like Post-it notes. So if you have Post-it notes, you can peel one off and peel another off, but you're not injuring the actual stack of Post-it notes. So that's kind of how our peripheral proteins are. They don't really damage the membrane if they get removed. Our different types of membrane proteins. We have anchoring proteins, which stabilize the cell's position, and they either attach to the inside or outside structures. Recognition proteins, which are identifying other cells and labeling cells as normal or abnormal. Glycoproteins are an example of recognition proteins. They're very important in immune function. If a cell is labeled as abnormal, then the immune system is going to come up and it's going to break down that cell and keep it from reproducing. That's how cancers begin, is through cells that have become abnormal. Enzymes help to catalyze reactions. Enzymes can be integral proteins or peripheral proteins. Receptor proteins bind and respond to ligands. A ligand is basically anything that can come and bind to these receptor proteins. So it could be an ion like calcium or hydrogen, or it could be a hormone or a neurotransmitter. All of those different things bind to receptor proteins. Carrier proteins with transport solutes through the membrane, and channel proteins which regulate water flow and some solute flow through membranes. We also have carbohydrates bound to our plasma membranes. These are proteoglycans, glycoproteins, and glycolipids. You'll notice that each one of these has the word glyco in it. Glyco refers to sugar. Sugar is a carbohydrate, so that's how you can remember. Proteoglycans, glycoproteins, and glycolipids are all membrane carbohydrates. They extend outside the cell membrane and form this sugary sort of sticky coat on the outside of the cell called the glycocalyx. The glycocalyx is going to lubricate and protect because it is that thick and sticky cell. It's lubricating. It anchors the cell, or it can also assist in locomotion. It has receptors for binding other things, so it would be binding different ligands. And when ligands bind, it can change the properties of the cell. And then also for recognition in the immune response, which helps us label cells as normal or abnormal. So that was our plasma membrane, which forms the outer layer of the cell. So now we're going to move into the cell and look at the different types of organisms or organelles within the cell. So our cytoplasm is all of the cellular contents between the plasma membrane and the nucleus. So cytoplasm contains everything within the cell aside from the plasma membrane and the nucleus. The components of this cytoplasm are the cytosol, which is the fluid portion of the cytoplasm. It's mostly water, but there are some dissolved solutes and things within the cytosol. The organelles, which are highly organized subcellular structures, and we're going to learn about each of the different types of organelles and what they do. And then inclusions, which are just masses of insoluble material and stored nutrients within the cell. The cytosol is also referred to as the intracellular fluid. So our organelles means little organs. We have two types, non-membranous organelles and membranous organelles. Non-membranous, as the name implies, have no membrane. So the components of those organelles are in direct contact with the cytosol. And these include the cytoskeleton, the microvilli, the centrioles, cilia, ribosomes, and proteasomes. And then our membranous organelles are organelles that are covered with their own plasma membrane that isolates them from the cytosol. And this includes the endoplasmic reticulum, the Golgi apparatus, lysosomes, peroxisomes, and mitochondria. Our organelles are responsible for performing most of the tasks required to keep a cell alive and functioning. If you're trying to picture what a cell might look like, you can think of a balloon. You blow up the balloon and then you fill the balloon with jello and fruit cocktail, so that the balloon itself is the cell membrane, and then the jello is the cytosol, and the fruit that is suspended within the jello are the little organelles. So I kind of like that analogy, so I like to share it. So we're going to look at each part individually. The cytoskeleton is just what it sounds. It's the skeleton of the cell. It contains structural proteins which give the cell its strength and its shape. Microfilaments are thin filaments composed of actin. Actin is a protein. The microfilaments anchor the cytoskeleton to the integral proteins in the membrane and determine the consistency of the cytosol. Intermediate filaments are mid-sized. They're made of collagen, which is a protein, and we find these in areas of increased stress. So places where the cell is stressed are going to have additional intermediate filaments. And then microtubules are hollow tubules made of a protein called tubulin, and they move vesicles within the cell, and they form the spindle apparatus. And we're going to talk about the spindle apparatus when we talk about cellular division. So here we have centrioles, which form the spindle apparatus. So it's made up of microtubules, and they surround the around the nucleus, and so when our cell divides, they help with pulling the chromosomes apart. And again, we're going to learn about that later, so I wouldn't worry too much about this slide right now. Microvilli are little extensions of the cell membrane, which increase the surface area for absorption. And they attach to the cytoskeleton at the terminal web. And when we come to another picture, I will show you what that looks like. We find microvilli a lot in the digestive system, where we are trying to absorb all of our nutrients from the foods that we eat. So we have all of this increased surface area so that we can absorb absolutely as much as possible. Cilia are often confused with microvilli. Cilia are small hair-like extensions off of the surface of a cell, and they beat to actually move fluid across the surface of the cell. So we find cilia within the respiratory tract that tries to beat mucus up out of the lungs. We find it within portions of the reproductive tract, carrying the egg from the ovary to the uterus. We have cilia that beats in the uterine tubes. So as you go through A&P2, mainly you're going to learn a lot more about these cilia and microvilli, but we will touch on them again this semester. And then a flagella is a single whip-like extension of a plasma membrane, and we only find the flagella in sperm. It is just one single whip. It gives the sperm its motility, and again, the sperm is the only cell in the human body that has a flagella. So here's a picture of the cilia, and you can see it's made up of microtubules in here. And so the cilia they do what's called the power stroke. So they stiffen, and then they only beat in one direction. And then once they've made that power stroke, then they lose their stiffness, they become flaccid, and they come back into this initial position where they stiffen again and then beat into that same direction. So they're always beating in one direction. So in the trachea, the cilia are always beating up toward the mouth, trying to keep mucus and debris and other things from entering the lungs. Because when you get things stuck in your lungs, you can end up with a lung infection. So the cilia are always beating in one direction away from the lungs. Same with the reproductive system, always beating to carry the egg in one direction. So this is how they do that, through the power stroke where they are firm, and then the return stroke where they are loose and not creating any energy to bring anything in the wrong direction. Proteosomes contain enzymes, which are proteases. They are proteolytic enzymes, which means that they are used to break down proteins. And so that's what they function, to disassemble damaged proteins, and then proteins get broken down into individual amino acids, and amino acids can be recycled and then used to assemble new proteins. So our proteosomes contain proteolytic enzymes or proteases, which disassemble damaged proteins for recycling. Ribosomes are sort of the opposite. They use amino acids to build proteins. So ribosomes are very important in protein synthesis. We have two different types of ribosomes in our cells. Free ribosomes, which are within the cytoplasm, and they manufacture proteins to be utilized within the cell. And then fixed ribosomes. Fixed ribosomes are attached to the endoplasmic reticulum, and they manufacture proteins that are going to be secreted out of the cell, so packaged and expelled from the cell to go and be utilized elsewhere. So free ribosomes manufacture proteins for the cell. Fixed ribosomes manufacture proteins that are going to be secreted out of the cell. We find a lot more ribosomes in tissues that require a lot more proteins. So the liver is an example. The liver produces most of our proteins that are in our blood. And so the liver has a lot of ribosomes because one of its main functions is packaging these, producing and packaging these blood proteins. The endoplasmic reticulum is a network of intracellular membranes that are continuous with the nuclear envelope. We have two different divisions of our endoplasmic reticulum. our rough endoplasmic reticulum, which is covered with ribosomes, and you can see that here, and our smooth endoplasmic reticulum, which has no ribosomes. They are filled with these cisternae, which are chambers within the membranes, and their functions are the synthesis of proteins. That's going to be our rough endoplasmic reticulum, because we know that our ribosomes are creating proteins. And then our smooth endoplasmic reticulum is going to secrete or assemble, synthesize carbohydrates and lipids. They also store what they create prior to being transported within the endoplasmic reticulum, and then they can be transported to the Golgi apparatus, where they can be packaged and secreted out of the cell. The endoplasmic reticulum also functions in the detoxification of drugs and toxins. The smooth endoplasmic reticulum has no ribosomes, so therefore it synthesizes lipids and carbohydrates, phospholipids and cholesterol, which are utilized in the cell membrane, steroid hormones, which are utilized in the reproductive system, glycerides, which are stored in the liver and fat cells, glycogen, which is stored in muscles, and then also functions in the detoxification of drugs. Our rough endoplasmic reticulum is covered with ribosomes. These are active in protein and glycoprotein synthesis. They fold polypeptides into protein structures and then enclose those in transport vesicles to be sent out of the cell and utilized elsewhere. The Golgi apparatus is a series of membranous plates called, they say it's a stacked dinner plates. We'll see that on the next slide. And what the Golgi does is it creates lysosomes and secretory vesicles, which are vesicles that can release things to the outside of the cell. Major functions are modifying and packaging secretions for release through exocytosis. So whatever this one cell is in charge of making, that can be packaged and then released through the Golgi. It renews or modifies the plasma membrane through membrane renewal vesicles, I believe. Packages special enzymes within vesicles for use within the cytoplasm. And it can add or remove carbohydrates to or from proteins to change a protein's structure and function. So here's a picture of our Golgi apparatus. Vesicles from the endoplasmic reticulum, these transport vesicles. Oh, no. Okay, I did not mean to touch that. I'm sorry. This does not let me go back. So the vesicles enter in the forming phase, and then they are processed through the Golgi and released in secretory vesicles by the maturing phase. Our secretory vesicles modify and package products for exocytosis. The Golgi also produces membrane renewal vesicles, which add or remove membrane components, and then it produces lysosomes, which carry enzymes to the cytosol. So here we are at our lysosomes. So lysosomes are enzyme-containing vesicles. Lyso means to dissolve, soma is the body. So lysosomes have powerful enzymes that can dissolve other organisms or can actually rupture and dissolve an entire cell. A primary lysosome is formed by the Golgi apparatus and contains inactive enzymes. So a primary lysosome is inactive. A secondary lysosome is when a primary lysosome fuses with a damaged organelle or a bacterium or something that has entered the cell and doesn't belong there. It's going to release its enzymes into this new vesicle where it has, let's see, so I'm looking here. So we have the damaged organelle up here. The primary lysosome is going to fuse to that damaged organelle and become a secondary lysosome. And then it's going to break down that organelle. We're going to resorb some of the things from that organelle that we could maybe reuse. And then the waste product is going to get released out of the cell through exocytosis. And we'll talk about what exocytosis is a little bit later. But this is how our lysosomes work. So primary lysosomes are inactive. Secondary lysosomes are going to fuse with a damaged organelle or some type of pathogen and digest that, and then resorb what we can utilize within the cell, and then release anything extra through exocytosis. Our lysosomes function to clean up inside cells. So they're breaking down large molecules, attacking bacteria, recycling damaged organelles, and then ejecting waste products by exocytosis. And they can also function in what's called autolysis. Auto is the breakdown of a cell itself. So when a cell is damaged beyond repair, the lysosomes are going to dissolve their membranes, release their digestive enzymes into the cell, and they can create death of the entire cell. So the whole cell is going to decompose. Peroxisomes are utilized to break down fatty acids and organic compounds. They produce hydrogen peroxide, but then that is later broken down by other enzymes, so it doesn't actually damage the cell. We have most of our peroxisomes are in cells that are very metabolically active, and peroxisomes are produced by cell division, whereas lysosomes are produced in the Golgi apparatus. So there's a little bit of a difference there between peroxisomes and lysosomes. So peroxisomes are produced by cell division, and they carry different enzymes than lysosomes. So their enzymes are just to break down fatty acids and organic compounds, whereas lysosomes can break down just about anything. Next, we have our mitochondria. Mitochondria are the powerhouse of the cell. They have a smooth outer membrane and then a folded inner membrane. So they have two membranes, smooth outer membrane and a folded inner membrane, and those folds are called cristae. The matrix of the mitochondria is the fluid that surrounds that cristae. And what the mitochondria does is they break down glucose and produce ATP. ATP is what our body uses as energy to power everything that we have to do within our body. And so we skipped chapter 2. You may want to look back quickly at chapter 2 and just see about ATP production and the Krebs cycle. You can also watch the crash course video on ATP. It's really helpful in just learning what ATP is. We're going to talk a lot about it. And in AMP2, you're going to learn about energy production toward the end of AMP2 with cellular respiration. But for right now, just know that ATP is our energy molecule within the body. It's how we power everything that we do, and it's produced in the mitochondria, most of it. The nucleus is the largest of our organelles, and it contains our genes. So all of the genetic code that tells all of our cells what to do, what they're going to be, how to make all the proteins, all of that stuff is contained within the nucleus. The nucleus has a membrane called the nuclear envelope, which is actually a double membrane that surrounds the nucleus. The space in between the two layers of that nuclear envelope is called the perinuclear space. And then there are these little pores through that membrane called nuclear pores, which allow for communication. And that becomes important when we start to talk about protein synthesis and protein synthesis. The nucleus contains our DNA, which is all of the information needed to build and run our organism. So all of the genetic material of our body is contained within the nucleus. The nucleoplasm is the cytoplasm within the nucleus. It's so special it has its own name. The nuclear matrix are the support filaments that give the nucleus its shape. And nucleoli are related to protein production. They're made of RNA, enzymes, and histones. Histones are proteins that DNA wraps around. And the nucleoli synthesize ribosomal RNA and ribosomal subunits. So that's what I want you to know about the nucleoli. They synthesize ribosomal RNA and ribosomal subunits. And we're going to learn more about that when we talk about protein synthesis. Nucleosomes are DNA coiled around these histones. So here you can see the histones are those little blue proteins, and then our DNA wraps around those, and so the DNA wrapped around the histones is termed a nucleosome. Chromatin is loosely coiled DNA. You can see that in this picture. And we find that in cells that are not actively dividing. And then chromosomes are tightly coiled DNA that we find in actively dividing cells. So this is all the different ways that our DNA can be made up. DNA contains the instructions for every protein in the body. Again, DNA contains the instructions for how to build every protein in the body. A gene contains the instructions for one single protein. So DNA, every protein in the body. A gene, one single protein. Our genetic code is a chemical language of DNA instructions, and it uses a sequence of bases called A, T, C, and G: adenine, thymine, cytosine, and guanine. Those are our bases within our genetic code. 3 bases, so our triplet code is 3 bases code for one single amino acid. Amino acids are the building blocks of proteins. So if you were to put A, T, C, that is the code for one amino acid. And then you would code for a second amino acid and a third, and then when those all get strung together, it's going to form a protein. So that is how we create our proteins, utilizing the genetic code within our genes, which are part of our DNA. You may want to listen to this slide a few times. Protein synthesis. The nucleus contains chromosomes. Chromosomes contain the DNA. The DNA contains the instructions on how to build the proteins. So proteins determine a cell's structure and function. So our DNA is going to have certain genes that are more dominant in certain cells that'll produce different proteins that'll give our cells their sort of final instructions on what kind of cell they're going to be, what they're going to do, and what they're going to produce. Protein synthesis involves 3 steps. The first step is transcription, which is where we copy the information or the instructions from the DNA onto a messenger RNA, and this takes place within the nucleus. So the DNA instructions are copied to a messenger RNA. So we're just making a copy of the instructions to build a protein. Translation occurs outside of the nucleus in the cytoplasm. And this is where a ribosome is going to come and read the code from the messenger RNA and then utilize that code to assemble our amino acids into polypeptide chains. So if you remember the genetic code, in triplicate code, we have 3 bases that code for 1 amino acid. So in the nucleus, the first thing that happens is we are making a copy of the instructions from the DNA into the messenger RNA. Then the messenger RNA is going to leave the nucleus and go out into the cytoplasm. And so now the messenger RNA is in the cytoplasm and it contains the copy of the DNA instructions. Now that it's in the cytoplasm, a ribosome can come, read the instructions in triplicate code, and it can begin to pull in the necessary amino acids that are going to get linked together to create the protein that we have the instructions for. So that's step 2, translation. Step 3 is processing. So once we have this polypeptide chain that got created in step 2 with the ribosome, that polypeptide chain gets sent to the rough endoplasmic reticulum and or the Golgi apparatus, where it is processed and then produces the actual protein that we're looking for. So transcription, copying the instructions from the DNA onto the messenger RNA. The messenger RNA then leaves the nucleus and goes out into the cytoplasm. Translation, the ribosome is reading the code from the messenger RNA and assembling the required amino acids into a polypeptide chain. Once that polypeptide chain has been made, it gets sent to the Golgi apparatus where it is processed and packaged into the protein that we desire. So that's step 3, is processing. So we're going to look at this in some pictures. So the first thing that happens is that our DNA strand is going to separate. So here you can see our DNA double helix, which is sort of wound around itself. And so that is made up of 2 strands here. So the one of them is the template strand. So the template strand is the strand that we're going to copy. And the other one is the coding strand. So the coding strand contains the code, and the template strand contains sort of the opposite of the code, or the mirror image of the code. So the first thing that happens is gene activation. And gene activation occurs when this protein, RNA polymerase, which is an enzyme, is going to separate the 2 strands, the coding strand and the template strand, at the what's called the promoter gene. So the promoter gene is the first gene that starts the signal to say this is the instructions for this protein. So then what happens is the RNA polymerase, after it binds to the promoting gene, it starts to move down the template strand. And at each site, the complementary RNA nucleotides are going to bind with the DNA nucleotides of the template strand. So that's happening here. So our nucleotides, our bases, are adenine, thymine, cytosine, and guanine, and they have complementary bases. So A and T are complementary bases, and C and G are complementary bases. So anywhere that you see a T, like here, then you're going to have an A come in and bind there, because A and T go together. And here you have C and G go together. So anytime So once we're copying the template strand and we are getting the complementary bases to come in and bind with the template strand, we're forming this mRNA, messenger RNA, and the messenger RNA now contains the code that the coding strand had. I'll try that again. So the DNA has two strands, a coding strand and a template strand. The coding strand contains the code to manufacture proteins. The template strand contains the complementary code. So adenine and thymine are complementary, cytosine and guanine are complementary. So the RNA polymerase is going to come in and bind at the promoter gene. So the promoter gene is like the start codon. It says this is where this instructions for this protein begin. And then as it comes down and moves its way down the template strand, there are the complementary bases are going to come in and bind, and they're going to form this messenger RNA. So the complementary bases for thymine is adenine, for cytosine is guanine. There is one exception here. Adenine and thymine are complementary bases. However, thymine is too large to fit out of the nuclear pore. So at this step, the thymine gets substituted with uracil. So instead of thymine coming in to be the complementary base for adenine, uracil is brought in. The uracil is much smaller. The uracil can fit through the nuclear pore. And so this mRNA strand that we end up with contains the code of complementary bases that have been attached here to the template strand. And so now the mRNA will have the same code as the coding strand, except it will have uracil substituted for thymine. So we just created our messenger RNA within the nucleus by bringing the complementary bases to attach to the template strand, and then they get released once they've copied the entire code. So the mRNA contains the code for creation of a protein. However, it has uracil substituted for thymine. That makes it small enough to leave the nucleus through the nuclear pore and enter into the cytoplasm. Once it's in the cytoplasm, a free ribosome, which is surrounded by amino acids out in the cytoplasm, is going to bind to the messenger RNA. Once it binds, then transfer RNA or tRNA molecules are going to bring amino acids and deliver them to the messenger RNA. So let's look at this in pictures. So first, translation. We have the mRNA strand, which is down here, is going to bind to a small ribosomal subunit. So that's what we're seeing in tan. This is the small ribosomal subunit. So it binds to the mRNA, and the mRNA contains the code. The start codon is the first triplicate code of the mRNA strand, saying start here and assemble your amino acids in this order. So a codon contains 3 bases. That's our triplicate code. So here our bases are AUG. So the anticodon is going to have the complementary bases to the codon. So we have AUG, so the anticodon is UAC because adenine and uracil are complementary, uracil and adenine are complementary, and then guanine and cytosine are complementary. Remember ATGC, T gets substituted with U. So the anticodon comes and binds to the codon like this. And the anticodon is attached to the transfer RNA. So the transfer RNA is a molecule that contains the anticodon, and it brings an amino acid. So it's attached to one amino acid, and this is the code for that amino acid. So when we call for this code, we are calling for this one specific amino acid. So once we have that first tRNA bound, we move down and we bind the second tRNA. So the second one, the second codon has a different code, CCG. So the anticodon is GGC, and GGC codes for a different amino acid than our UAC coded for. So here's one amino acid and two amino acids. You can see that they're different colors, so they are different amino acids. So now we have two transfer RNAs that have brought in two amino acids. Then our ribosomal subunit or ribosome now is going to move down the mRNA strand, and the first tRNA is going to get released, but it's going to leave behind its amino acid bound to the second amino acid through a peptide bond. A peptide bond is a bond between two proteins. So now we have two amino acids that are strung together or connected together here, and this transfer RNA is still connected to the ribosome. So now we have a third transfer RNA with the anticodon that is complementary to the third codon on our messenger RNA strand. So the third tRNA is going to come and bind. The ribosome is going to move down, and the second tRNA is going to leave, and we're going to then bind all three of those amino acids together. Once the ribosome has worked down the entire messenger RNA chain, it will have assembled all of the amino acids required to create that one protein. Then the two ribosomal subunits are going to release from one another, and this polypeptide chain is going to be sent to the Golgi apparatus for processing. So here's our triplicate code. Please do not memorize these individual amino acids and what their codes are. I like to put this slide in so that I can show you that each amino acid has its own specific code. So phenylalanine is one amino acid. Its code is thymine, thymine, thymine. So the template strand, which is complementary to the code, would be adenine, adenine, adenine. When the mRNA is created from the template strand, the uracil is substituted for thiamine. So the mRNA codon is uracil, uracil, uracil. And that would call in the anticodon on the transfer RNA of adenine, adenine, adenine. And that tRNA with the three adenines is the code for phenylalanine. So it holds the phenylalanine amino acid. So if we're reading the mRNA, and the codon is UUU, that's going to call in the tRNA that's holding one single phenylalanine amino acid. Then the next codon could be GCC, right? So the GCC would call in the tRNA with CGG anticodon, and that would contain the alanine amino acid. So if this was your first one and this was your second codon, then you're going to end up with a phenylalanine attached to an alanine. Then you could add a cysteine, you could add a methionine, and then you could add a proline. So any of these chains of amino acids are going to create different proteins. So that's how our triplicate coding works and how we are coding for calling in amino acids to be built in a specific chain and then processed into a protein. All right, so you may want to rewatch that whole portion of the lecture 2, 3, 14 times. Try to really wrap your head around how all of that works. There are also some videos online that you can watch, and certainly read that portion of your textbook to make sure that you're understanding what I'm talking about. So now we're going to move on to membrane transport. So the cell membrane is a barrier to nutrients and waste products. However, nutrients need to get into the cell and waste products need to get out of the cell, as well as products that the cell is going to be producing, such as hormones, proteins, etc. Permeability of the cell membrane is what determines what gets in and out of the cell. We have 3 different types of permeability. Impermeable means nothing is allowed in or out of the cell. Freely permeable means anything can pass in and out of a cell. And selectively permeable means some things can move freely and other things cannot. And our cells are a selectively permeable membrane. Permeability is selective, and it can restrict materials based on their size. So are they too big to fit through a channel? Based on their electrical charge. We have an electrical charge within our cell across our plasma membrane, and so like charges are going to repel one another, opposite charges are going to attract. The shape of the molecule and lipid solubility. So if it is fat soluble, it can diffuse through the plasma membrane. If it is water soluble, it cannot. We have active transport, which requires energy in the form of ATP, and then passive transport, where no energy is required in order to transport specific things across a membrane. So we have 3 different mechanisms of membrane transport. Diffusion, which is passive, carrier-mediated transport, which can be passive or active, and vesicular transport, which is active. So again, remember, active means requires ATP. This would be a good time to watch the second crash course video. There's a crash course video specifically on membrane transport. All of our molecules in our body are constantly in motion, so they're always moving, and they move in a random pattern. Random motion causes mixing of different substances. And so we're going to talk a little bit about just general concentration, diffusion, things like that. So the concentration is the amount of solute that is within a solvent. So the solute would be like particles that are within a solvent. The solvent would be the liquid. A concentration gradient is where you have more solutes in one part of a solvent than another. So if you were to take a, here we'll look at the next slide. So here we're going to look at diffusion. So diffusion is a function of the concentration gradient. So we're taking this solid and we're dropping it into water. So now we have a high solute concentration at the bottom of the solution or the solvent, and a low, there's no solutes up at the top of the water. So we have a very different concentration gradient here, right? We have a lot of solutes at the bottom, no solutes at the top. So those solutes are going to want to diffuse because they want to eliminate a concentration gradient. And because all of these ions are constantly in movement, they eventually are going to spread out until they reach equilibrium, which is where the entire solution has the same concentration as the rest of the solution. So you have equal solutes in every part of the solution. So this is diffusion, where you have a high concentration of solutes, and those solutes are going to spread out to eliminate a concentration gradient. And that's what nature always wants to eliminate concentration gradients. We have many different factors that affect diffusion. So the distance the particle has to move, the farther the distance, the longer it takes for diffusion to occur. The size of the molecule, small molecules move faster, large molecules move more slowly. Temperature affects greatly, and when we when we are in class together, we do a fun lab experiment on temperature. So the hotter a solution is, the faster the diffusion occurs. The colder it is, the slower it occurs. The gradient size, so the difference between the high concentration and the low concentration, a larger gradient is going to change more quickly. And then the electrical forces, opposites attract and like charges repel. Diffusion across a plasma membrane can be simple diffusion, such as lipid-soluble molecules, which we can see right here, which can just diffuse directly through the plasma membrane because they can get through that hydrophobic tails of the phospholipid bilayer. Or channel-mediated diffusion, which is utilizing carrier proteins, so that here we've got some water molecules and small ions that can diffuse directly through these open channels. Osmosis is the term for the diffusion of water across the cell membrane. So remember, when we're talking about diffusion, we're talking about the movement of solutes. Osmosis is the movement of water. The more solute molecules there are, the less concentration of water molecules. Because if you have a specified amount of space and you have more solutes, that gives you less space for water. In order for osmosis to occur, you must have a membrane that's freely permeable to water and selectively permeable to solutes. The water molecules are going to diffuse across the membrane toward the solution that has more solutes, trying to dilute that solution. Therefore, the volume is going to increase on the side with the more solutes. So let's look at this in pictures to make it make more sense. So here we've got a test tube. We've got water on both sides, and the level is going to be the same on both sides of like total amount of water plus solutes. And then there's a selectively permeable membrane in the middle. So you can see on the left side, we have fewer solutes and more water. And on the right side, we have more solutes and less water. So naturally, the water is going to want to move from the side with more water to the side with less water so that it can dilute the solution on the right. So we end up with a greater amount of water on the right-hand side so that we can have an equal number of water and solutes on both sides. So hopefully you can see how that happened, that the water had to move over to dilute the solution on side B in order to have the same concentration in side A and side B. Osmosis can be prevented by resisting a change in volume, which would be having to apply a force onto side B to keep that water from being able to move over, and that is osmotic pressure. Osmolarity is the term for the total solute concentration in an aqueous solution. Aqueous means water-based, so osmolarity is the total number of solutes within a watery solution. Tonicity describes how a solution is going to affect a cell, and this becomes very important within the body. The nature of solutes is often as important as the total osmolarity. So two fluids can have equal osmolarity but different tonicity. And we have three types of tonicity that I want you to pay attention to. Isotonic means that there will be no net change of fluid within a cell. So you'll still have fluid coming in and fluid going out, but no net change. So you can drop a cell into an isotonic solution and nothing will happen to that cell. Hypotonic means the solution has fewer solutes than the cell, so the water that's in the cell is going to want to leave the cell to dilute the solution around it. And hypertonic means the solution has, I'm sorry, I just gave you the hypertonic explanation. Hypotonic, the solution has fewer solutes than the cell, so the solution is going to lose water and the water is going to go into the cell to try to dilute the cell. In a hypertonic solution, the solution has more solutes than the cell, so the cell is going to lose water, the solution is going to gain water through osmosis because the water is going to want to leave the cell to dilute the solution. So let's look at how this affects a red blood cell, because we always want to, sometimes we have to talk about these larger concepts, but we like to put them in context of the actual body. So here we've got a red blood cell. Red blood cells are biconcave discs. They look like that. So in an isotonic solution, you drop the red blood cell into this isotonic solution, nothing happens. You have water moving in, water moving out, but the red blood cell does not change. In a hypotonic solution, we have more solutes within the red blood cell. You can see we have 4 solutes here within the red blood cell, no solutes outside. So the water is going to want to enter the cell to dilute those solutes. And that can actually cause a cell to rupture. Hemolysis or hemolysis is the rupture of a red blood cell. And so that can happen in a hypotonic solution. In a hypertonic solution, we have a lot more solutes outside the cell, fewer solutes inside the cell, so the water is going to want to leave the cell to dilute the solution, and then the cell is going to undergo what's called crenation, which is the shrinking or shriveling of a red blood cell. So when you rupture a red blood cell, it can no longer function. When a red blood cell shrinks or crenates, it can no longer function. So it's important that our body is always full of these isotonic fluids so that we can still function. Our red blood cells carry oxygen to all of our tissues, and they're extremely important. So if they cease to function, then very quickly our entire body ceases to function. So carrier-mediated transport, so we were just talking about diffusion and osmosis. Now we're going to talk about carrier-mediated transport. So these transports ions and organic substrates through facilitated diffusion or active transport. Characteristics of these are specificity. So there's one specific transport protein that will move one set of substrates. For example, we have the sodium-potassium exchange pump. So that one protein will move sodium and potassium and nothing else. Saturation limits, the rate depends on the transport proteins. So if the proteins are moving as much as they possibly can, then the amount of substrate in the area doesn't matter. It really depends on the number of proteins and their ability to move substrates. And then these are often regulated by cofactors such as hormones, which need to bind to those proteins and allow those proteins to function. We have two types of carrier-mediated transport, co-transport and counter-transport. In co-transport, we have two substances that are moving in the same direction at the same time. So our two substances are either moving both into the cell or both out of the cell. Whereas counter-transport is going to be moving substrates in opposite directions. So you can move one substrate into the cell while you're moving another one out of the cell. Facilitated diffusion is passive. It involves carrier proteins that are going to transport molecules that are too large to fit through channel proteins. So our channel proteins are always open and they allow certain ions to just pass through. But some ions are too large to pass through those channel proteins, so they require carrier proteins. So in this instance, we're looking at the transport of glucose. So the glucose is going to bind to the receptor within the transport protein. That causes the protein to change its shape, and then it's going to drop the glucose into the cell. So this is facilitated diffusion. In active transport, we can transport substrates against their concentration gradient, and this requires energy. So here you can see an ATP is being spent. ATP, when it gets utilized, it gets broken down into ADP. So if you see ATP to ADP, that means one ATP is being utilized. And in this instance, this is the sodium-potassium exchange pump. So we are pumping 3 sodium out of the cell, even though there are more sodium outside the cell than inside the cell. So sodium is going against its concentration gradient. And we are pumping 2 potassium into the cell, even though we have additional potassium, more potassium inside the cell than outside the cell. So we're utilizing energy to move substrates against their concentration gradients. And this is an example of counter transport because the sodium is leaving the cell and the potassium is entering the cell. So the two substrates are going in opposite directions, which is counter transport. Secondary active transport happens with, here we have sodium concentration gradients that are driving glucose transport. So we've got a transport protein where there's a glucose receptor and there's also a sodium receptor. So the sodium binds and the glucose binds. That allows this protein to change shape and it's going to drop the glucose and the sodium into the cell. However, later on, this sodium has to get transported back out of the cell utilizing the sodium-potassium exchange pump. So it's called secondary active transport because we don't need ATP in order to get the sodium into the cell. The sodium brings the glucose along for the ride, but we do need ATP to pump the sodium back out of the cell. Vesicular transport utilizes vesicles, also called bulk transport, to move materials in and out of the cell. So endocytosis means bringing materials into the cell, and this can happen through receptor-mediated using ligands. Ligands are going to bind to receptors on the cell membrane. We'll see that in a picture. Pinocytosis or phagocytosis. Pinocytosis is cell drinking, phagocytosis is cell eating. And then exocytosis is the release of things out of the cell. So it can be granules, droplets, packaged products can all be released through exocytosis. And we'll learn a lot about exocytosis when we learn about transmission of nerve impulses at the synapse and how neurotransmitters are released through exocytosis. So that's coming later. So here's our receptor-mediated endocytosis. So here are receptors, all of these little receptors you can see. This one and this one are already bound to these ligands. So the ligands are whatever molecules the receptors are sensitive to. So the receptors are going to bind to all of these ligands, and then the ligands are going to, this whole part of the membrane is going to pinch off, and so it's bringing all of these ligands into the cell. So now you have this vesicle and it's full of all of these ligands. So a primary lysosome is going to come and fuse to that vesicle, creating a secondary lysosome. The secondary lysosome is going to use its enzymes to release all of those ligands from their receptors. And then the ligands are going to be removed from the vesicle so they can be utilized within the cell. So say we needed, I'm not even sure. I don't want to make something up here, but substance A. Okay, so we need substance A in our cell to make our cell function. So these receptors would be substance A receptors. So when enough of them bind, it creates this vesicle. The lysosome then releases the substance A from the receptors and releases the substance A into the cell so that we can use it. And then the empty vesicle goes back and attaches back into the cell membrane. So all of those receptors are back there, and then the whole process can start all over again. Here's a picture of pinocytosis, which is cell drinking. So you have vesicles forming within the cell membrane, and they're taking the fluid that's outside the membrane and bringing it, pulling it into the cell. Phagocytosis is cell eating. So in this way, we are bringing a pathogen or a bacteria into the cell, and it is being broken down by lysosomes. The lysosomes you can see here are being formed in the Golgi apparatus. The lysosome is going to attach to the vesicle that contains the pathogen, and then it's going to break down that pathogen, and the contents will then get released outside of the cell when they are now inactive and cannot harm the body. So this is how a lot of our immune cells function. They are phagocytic cells, which means they roam around your body looking for bacteria, and they attack them by pulling them into the cell and then dissolving them with their lysosomal enzymes. And here's a picture of exocytosis. So in exocytosis, it's sort of the opposite. We have a secretory vesicle that's full of a substance that we want to release from the cell. So that vesicle is going to fuse to the plasma membrane and then open up and release its contents out of the cell into the intercellular fluid. We're going to do a very brief introduction to transmembrane potential, because next time you hear about it, I don't want you to feel like you've never heard about it. Transmembrane potential becomes extremely important when we learn about the muscular system and the nervous system. So we're going to do our little intro here. So transmembrane potential is the difference in charge between the inside of the cell and the outside of the cell. Inside of each cell has a slightly negative charge, and outside of the cell has a slightly positive charge. The cell membrane separates the positive charge and the negative charge, and that separation creates a transmembrane potential. Remember that opposite charges attract, so the positive charge and the negative charge really want to get at each other. However, the plasma membrane is keeping them apart. So the resting membrane potential of a cell is when the cell is at rest, the change in charge from the inside to outside of the cell. And depending on the cell type, that can range anywhere from -10 millivolts to -100 millivolts. This becomes important because when you change that transmembrane potential, so you have all these positive ions on the outside and negative ions on the inside, and there are gates that would allow those positive charges to flow into the cell, mostly sodium in this instance. So if we do something to that plasma membrane where we allow the sodium gates to open, and then the sodium, the positively charged sodium ions outside the cell can rush into the cell, that can cause an excitement of the entire cell membrane, and then that can sort of bring information in the form of what we call an action potential. So again, we're going to really go in depth with this next when we talk about the muscular system and muscular contraction, and then even further when we talk about the nervous system. So I just wanted to give you a little, since we're talking about the cell membrane, transmembrane potential is extremely important within our cellular anatomy. So the last thing we're going to talk about is the life cycle of a cell. We're not going to spend a ton of time working on this, but our cells do divide, and so our life cycle is from when a cell becomes a cell until it divides and becomes two cells. So most of our cell's life is spent in a non-dividing state, which is interphase. But our cells do divide to create new cells, and that division happens in 3 stages. DNA replication, which is creating a duplicate of all of the genetic material within the cell. Mitosis, which is the division of the genetic material between from one cell to 2 cells. And then cytokinesis, which is the division of the cytoplasm of one cell into 2 cells, which are then termed daughter cells. So here's our life cycle. So interphase is all of this in purple, and that is our non-dividing state. And then mitosis and cytokinesis occur when a cell is ready to divide. Mitosis is made up of prophase, metaphase, anaphase, and telophase. You can read about those in your book. Let's see. Yeah, I guess we're going to get to it. Hold on. All right, here's our picture of DNA replication. So it's very similar to when we were talking about the creation of messenger RNA and having those complementary bases. There is also a crash course video specifically on DNA replication. So the first thing that happens is the helicase unwinds. So remember here we have the DNA unwinding, and by disruption of the hydrogen bonds, so we end up with these 2 strands. DNA polymerase, which is this enzyme here, is going to move its way. It's going to bind to the nitrogenous bases, and then it is going to move down the chain and call in the complementary bases, which will get linked by covalent bonds. So we are basically bringing in all of the complementary bases. So we're taking one set of DNA, and then we are going to make it into 2 separate sets of DNA. All right, so here's our one set. We're going to open that, and then we're going to bring in all of the complementary bases on each of the 2 strands, so we end up with 2 full strands of DNA. Here's a little video you can watch on mitosis. So the divided DNA, the duplicated DNA divides into 2 sets of chromosomes. Chromosomes are DNA that are coiled tightly into chromatids, and the chromatids connect in the center at what's called the centromere. And around the centromere, we have this protein complex called the kinetochore. During prophase, the nucleoli are going to disappear. The centriole pairs are going to move to the poles of the cell, and then the microtubules form the spindle apparatus, and this is going to extend between the centrioles. So it's between the two sides of the cell, and the spindle fibers are going to sort of push the edges of the cell away from one another. During metaphase, the chromosomes are going to align in a central plate called the metaphase plate. During anaphase, the spindle apparatus pulls the individual chromosomes apart and creates a set of chromosomes on either side of the cell. During telophase, the nuclear membranes are going to reform on either side of the cell, so you're going to have two nuclei within this cell surrounding each of our daughter chromosomes. And then our chromosomes are going to uncoil again. Our nucleoli are going to reappear. So we now have one cell that has two complete nuclei. Last is cytokinesis, where the cytoplasm itself is going to sort of involute in the center and then separate and produce two fully separate daughter cells. Our cells all divide at different rates. A slower mitotic rate means a longer cell life. So a cell only lives until it divides. So if it divides every 5 minutes, then the cell life is 5 minutes. If it divides every 5 years, then the cell life is 5 years. So the slower the mitotic rate, the longer the life of the cell. Cellular division does require ATP. So without ATP, our cells cannot divide. Muscle cells and neurons rarely divide. So that's an important bit if we were to say, what kind of cells divide? So muscle cells and neurons rarely divide, whereas exposed cells like skin cells and the cells in the digestive tract divide rapidly. Normally, we have a balance between cell division and cell loss. So we are creating new cells at about the same rate that old cells are dying. Don't worry about this part here. Repressor genes are genes that keep our cells from dividing or that decrease our cellular divisions. Telomeres are the terminal segments of the DNA. So every time a cell divides, the telomere is going to shorten. And when a telomere gets worn out, a cell is going to stop dividing. An oncogene is a mutant regulatory gene that can cause cancer. So if you have a faulty repressor gene, then that cell can divide and divide and divide, and every time a cell divides, there is a potential for something to go wrong, and when something does go wrong, eventually that can lead to a cancerous situation. Tumor is a neoplasm is a cancerous mass of enlarged like cells that have created a mass that grow abnormally and divide. So they're going to grow very, very quickly and divide pretty rapidly. A benign tumor is a contained tumor that is not life-threatening. So benign tumors, if they grow large enough, depending on where they're located, they may press on something and interrupt normal function and need to be removed, but they're not malignant. A malignant tumor is a tumor that can invade the surrounding tissues and or release cells into the bloodstream, which can then travel to distant regions of the body and create secondary tumors. So malignant cancers are much more dangerous. They have the ability to move throughout your body and create secondary tumors, and they're the ones that are much more life-threatening. Cancers can develop with a cell becomes abnormal. So here you can see that one cell has become abnormal. So something went wrong within that cell. Then that cell is going to produce a primary tumor, which is where it's going to rapidly divide and create daughter cells that all have this defect. And then that cell is going to send cells, or that tumor is going to send cells into the bloodstream. They're going to flow through the bloodstream and anchor into different areas of the body where they can create that secondary tumor. Talking about cancer, we could spend an entire semester just talking about cancer. And we'll find that a lot through A&P1 and A&P2. We're really focusing on teaching you normal anatomy, and we try to pepper in some of these what can go wrong, but it's taking a pathology class would be really fantastic in learning all of the things that can go wrong. So before you can know what can go wrong, you have to know what right looks like. So we're really focusing on what happens when everything goes right, with a little bit of this like, hey, if this goes wrong, this happens. So things to remember: every cell in the body carries your complete DNA instructions for all of your body functions. So every cell in the body has the instructions to create every protein in your body. Cells, however, differentiate. After they're formed, they can become any kind of cell: a liver cell, a fat cell, a nerve cell, by turning off genes that are not needed by that specific cell. So they still contain that genetic information, but they have specific genes that are turned off, and they're only utilizing the genes that a liver cell would need, or the genes that a neuron would need. All body cells, except for the sex cells, contain the same 46 chromosomes. And differentiation depends on which genes are active and which genes are inactive. So I know that was a lot of information. What I would want you to focus on for the beginning of the lecture is what are the different components of the cell and what is their major function. And then definitely spend some time on protein synthesis, because that is a challenge. And then on the different types of membrane transport, which ones use ATP, which ones don't. Know the difference between co-transport and counter-transport. And then know the phases of mitosis and sort of the main thing that occurs in each phase. All right, so that is the end of this week, and I will see you guys back here next week. I hope you have a wonderful week of learning.